Hi, this is Tom Salemi from OIS-TV. Happy to be joined by David Bailey, CEO of Sensimed. David, thanks for joining us.
Thank you. My pleasure.
Now, I understand you introduced Sensimed to our OIS conference today.
Yes, yeah, we presented the update, a couple of new things in there. First time we'd presented since we got the FDA approval in March. So that was exciting. And we also announced that we closed a $10 million convertible loan. First time we'd announced that,
Wow.
Of which half came from Seed, a Japanese contact lens company that wanted to get into the device space. So Seed came in with half of those monies. Our investors came in with the balance. And a portion of what Seed put in will be dedicated to doing a key pivotal study in Japan on using Sensimed triggerfish as a classification device for normal tension glaucoma.
Well, let's talk about triggerfish.
Two new exciting announcements. First time at OIS.
Thank you for sharing them at OIS. We love to break news here.
Yep.
Let's back up a bit and introduce triggerfish as well to our viewers out there.
Right. So triggerfish is the first FDA-approved contact lens sensor. So we have a sensing mechanism inside a silicon contact lens with a telemetry system so that we can record ocular volume change. So there's a strain gauge sensor inside the contact lens, and we measure the change in ocular volume over a 24-hour period so that we get an uninterrupted look at what's happening while the patient is asleep at night. So we get a full 24-hour measurement of something new, ocular volume change, which we're now focused on understanding the clinical relevance of that measurement and how it can be used to supplement IOP measurement to give better treatment for patients.
So is it clear that we need that second metric? Is it evident to you that that's necessary?
Excellent question. If you go to Japan, I think this is the key test. Mm-hmm. In Japan, because 97% of their patients have normal pressure, just measuring pressure alone for glaucoma diagnosis is not sufficient.
Oh,
they're desperate for an additional biomarker that they can use to supplement their IOP measurements in order to classify the patients more precisely and therefore decide treatment.
Is there generally a correlation, do you think there will be, between the two? Will someone who's not seeing increased pressure, will they likely register on your measurement, or is there any sort of connection between the two?
Yeah, the two, IOP and ocular volume change, we've shown are highly correlated, mm-hmm. but we can't simply calibrate our measurement straight to an IOP measurement. So I think in the future we see that the two measurements will be complementary pieces of information that the doctor will be able to use to give them a clearer picture of the kind of patient they're looking at. Mm-hmm. And the NTG classification is one area of focus for the understanding the clinical significance of the measurement. The other key one is we're really trying to push the envelope by trying to show that the measurement can be used to predict the course of the disease. Oh. And at the moment, all we do is watch and wait. Mm-hmm. And so I showed a paper that had been presented in Ophthalmology, which showed that certain elements of the triggerfish curve could be used to predict fast and slow progressors. And then I showed for the first time that we'd actually taken that to the next step by using a huge base of in-house data that we have of over 1,000 patients. We selected almost 450 of those patients, which was 10 times the size of the population in the ophthalmology study. And we repeated the analysis. In that cohort, which was from a registry, not from patients in a controlled study, so from 13 countries where you would expect a higher level of noise than you get from a clinical trial cohort. Mm. And we were able to once again show that elements of the triggerfish curve were highly correlated with the prediction of the disease progression, either fast or slow. So based on the one 24-hour measurement, with a degree of accuracy of over 80%, the doctor was able to say this is a patient where the disease will progress more quickly or more slowly. And we believe, based on our interaction with physicians, that that would be a very important thing to know.
Mm-hmm.
Okay?
How significant, we hear a lot about sensing contact lenses, obviously Google and those folks working on their projects. How significant is it to get an FDA approval of a contact lens sensor? It's different than what they're doing, but it's something.
I think the significance is that it shows Sensimed, it validates the Sensimed technology and the platform as a whole. And we took that through the de novo process. So I think the significance is it shows that a small company, Sensimed, is really leading the way in terms of contact lens sensing. And it shows that we have a platform approval. It's very easy to do a plug and play with a different sensor into our contact lens. So we have already taken a pure pressure sensor, put it into the same contact lens, and have used that in patients in studies. So I think the significance is it validates our technology as a small company and shows we have something that measures something that's repeatable, reliable, and in a safe way. And it gives us a platform approval from which we can expand. And at the same time, we're working on this clinical understanding in order to then expand the labeling. So I think it was really a milestone for Sensimed, particularly, as you say, when we were against these big players with all their big announcements. I mean, the only surprising thing, and I almost said this at the beginning of the presentation, is that I was stood up there not talking for Google or Alcon or J&J or Hewlett-Packard or CooperVision. You know, this is Sensimed, and we are truly leading the way with the first ever approval in this field. And I think that's good for everyone.
I agree completely, and we really do appreciate you sharing that news on the OIS stage.
It was my pleasure. A great meeting, and thoroughly enjoyed the interaction.
Thanks for joining us.
Thank you.